Hi, everyone. Thanks for joining us today. I'm Angie. I'll be introducing your presenters here in a moment, but while we're waiting for some people to join us, I'll just go over a few housekeeping tips. So hopefully you guys are here for the Shedding Light on Ocean Acidification webinar. That's what we'll be talking about today. Feel free to use the chat or the Q&A feature. We'd love to answer your questions during the session. And if we happen to not get to your question during the session, we will follow up with you afterwards. And you'll also get a link here in a moment to a shared Google folder that has the slide deck that Colleen's going to be going over, as well as some additional resources. And then I will also be putting in the chat that everyone who attends today will get a follow-up email with the link to the webinar recording, a certificate of attendance, and the link to that shared folder. So with that, I'm going to pass it on to Colleen and John.
Hi, everybody. Thanks for joining us on a holiday week here. Hopefully you all are enjoying it, and hopefully it's not going to be too crazy for you. So I'm going to go ahead and start sharing my screen here. So just to give you a little bit of agenda today. Today we're going to be, I'm going to introduce myself and John. We're going to be giving some background information on ocean acidification, and then we're going to do an actual lab activity and be able to share the data over the internet with y'all by using Graphical Analysis Pro. And then we'll have some time for question and answer at the end. So a little bit about myself. I have been at Vernier now for just over 8 years. My background is in general biology with a bit of a concentration, actually molecular biology. And I also have a master's in science and education from Montana State University. I actually used to be a research scientist and then got a grant to become a high school teacher, where I taught for 6 years in Houston, Texas, where I was lucky enough to teach freshman biology and AP environmental science, which in my opinion is the best class ever. So much fun to teach. And we have John joining us too here. So John, if you want to introduce yourself.
Hi, my name is John Melville. I have a BA in biology and psychology from Sonoma State University. I got my PhD in zoology from Oregon State University, and then did a postdoctoral fellow. But most importantly, I was a former college professor. I was an associate professor of biology at a small college called Wartburg College. And I taught lots and lots of different courses, including marine biology. So my original bachelor's degree from Sonoma State University actually had a large emphasis in marine biology. So that was part of my degree in biology, was with the marine emphasis and aquatic emphasis. And then I became a neuroscientist later. So.
All right. Thank you. So while we're doing this presentation, please don't hesitate to ask any questions. John and Angie are there to answer them or bring them to my attention while I'm talking and doing the demonstration. So we're going to try to answer them as we go. And then at the end, we'll have time to discuss some of those questions, or if there are other questions that pop as we go. In this experiment, we're going to be using our app called Graphical Analysis. I highly recommend going ahead and downloading it, because I'll be actually sharing my data with you all after we've collected some data here. And that way you can kind of see how you can have data that you can then share with students and have them be able to do their own analyses with that data. So let's talk a little bit about ocean acidification. So I just wanted to kind of cover some background information to make sure everybody was kind of on the same page of understanding. So the key thing to remember here is that when we're talking about pH, that pH is measured on a logarithmic scale. So when we talk about a change of pH from 7 to 6, for example, we're talking about a 10-fold change. So what that means is that any type of change we see in pH is going to be fairly dramatic because of that logarithmic scale. So any sorts of decrease or increase in pH is going to have a dramatic effect on the environment, because that pH in those environments is really meant to be pretty stable, especially in aquatic environments where they need to have that stable pH to make sure that certain organisms can live in the proper environment and have their proper niche needs met. So when we talk about ocean acidification, what we're referring to is looking at how the pH of the ocean has been changing. So currently, it's reported that the average pH of the ocean is it's a little bit on that basic end, where it's about a 8.1 pH. Now, it has decreased from a pH of 8.2. Well, we're talking about a 10th here. Remember, on the logarithmic scale, that's still going to be a pretty decent change. So the thing to keep in mind when we're talking about ocean acidification is the process here. We're looking at a natural process. So carbon dioxide is a gas that's just going to naturally be dissolved into any water, ocean water or fresh water. And what is expected is that about 30% of atmospheric carbon dioxide is actually absorbed by the world's oceans. So that's a lot of carbon dioxide being absorbed into the ocean. And when carbon dioxide gets absorbed, it's going to react, and this is the reaction I have right here. It's going to react with the water in the ocean and form carbonic acid. And then very quickly, that carbonic acid is going to dissociate into hydrogen ions and bicarbonate ions. And this is just a completely natural process that takes place. And we kind of don't worry about it too much typically, because the ocean has this natural, what we consider buffering ability, because of the ions and everything within the water. Now, the problem is when we think about things in relationship to climate change, that as we're seeing with climate change, our climates are changing due to the increase of carbon dioxide in the atmosphere. And what we want to make sure we're referring to is this excess amount of carbon dioxide. It's not the normal amount that gets absorbed into the ocean. We're talking about more than normal. And so because more carbon dioxide gas is being absorbed into the ocean, this is actually having a big effect on ocean chemistry. And what we're seeing is that the pH of the ocean has started to decrease. And so the question is, why is this so important? Well, what it really comes down to is by changing the pH and the chemical reaction taking place of forming carbonic acid, this reaction is also actually sucking up a lot of carbonate ions that are present in the ocean that different organisms need to survive. And so by adding more and more CO2, we're getting more and more bicarbonate being produced and making less carbonate actually available. And carbonate's actually really, really important in the ocean ecosystem, primarily because it builds a lot of the building blocks of bones and skeletons of the different organisms. So we talk about how that's going to create these bony structures on the outside of these organisms. And primarily we look at plankton and coral, because they are lower on that food chain, they're forming that base. And so if they have less carbonate ions available in order to build their skeletons, we're essentially seeing what we would consider an osteoporosis of the sea, where they're not able to build these really strong, robust structures in order to help basically live and support the rest of the food chain that relies on that. This is a great graphic here I took from NOAA that has the reaction that we're talking about here, and it actually has some images showing the effect on a specific type of plankton that's found in the ocean. And so this is why ocean acidification is of very high concern and importance for us to understand, because of the effect it's going to have on oceanic food chains and the resulting problems that could cause. So In our experiment, we're going to be doing an ocean acidification type experiment where we're going to see what happens when we add carbon dioxide to water samples and what's going to happen to the pH of those samples. So in a classroom setting, we can't exactly be looking at, in most situations, marine organisms and expose them to different amounts of acid and see how it affects them. What we can do is help students understand the process of adding excess CO2 to water samples and how that's going to affect the pH of that sample. So for this experiment, you need to have a source of excess carbon dioxide. And for the demonstration I'm doing today, I'm actually just using a basic over-the-counter Alka-Seltzer tablets. So in Alka-Seltzer, what we have here is sodium bicarbonate and citric acid. And when we put these in a water, what they're going to produce is sodium citrate, which is what people are wanting to help settle an acidic stomach, but also it's going to produce carbon dioxide. So that's going to be our main source of carbon dioxide for this experiment. You can also use dry ice if you would prefer. It definitely is a lot more fun for students to play with dry ice than Alka-Seltzer, but there are some kind of lab hazards you have to take into consideration with dry ice because it can very easily burn skin and it can build up pressure quite extreme in Erlenmeyer flasks that we're going to be using. So I don't want to encourage breaking of those flasks. Okay, so let's change what I'm sharing here. Oop. Share. Okay, so I'm going to be using our graphical analysis software, and I'm going to change my camera view if you can see me here. And so we're going to be using just our Go Direct pH sensor, and you can use any sort of pH sensor for this. I don't recommend using any pH paper because it's going to be a very quick reaction that happens. And the pH paper is not going to be able, it's going to give you kind of a beginning and an end, but it's not going to be as sensitive as, say, a pH probe would be. So I've turned on my pH probe, and in graphical analysis, I'm going to start my sensor data collection. And I'm going to wait for my pH probe to pop up here. Connect. And done. And I'm going to change my view a little bit to help make it a little easier for everybody to see. Go to 1.25, and then I'm also going to put up my meter so that's a little clearer there. All right, so what's great about this experiment is it's super quick and fast. All you need is the Erlenmeyer flask, some different water samples, a pH probe, and then what I use is a stopper with a tube attached to it. And this is going to be where I actually feed my carbon dioxide being produced by this reaction into my water sample. So first order of business, I'm going to take my pH probe out of this buffer solution. And the pH probe I've been having sit in a pH 7 buffer solution. And that's important because pH probes are normally stored in about a pH 4 buffer. So by using a pH 7, this is going to help get the starting pH a little bit more on level with what our sample is going to be and be able to have a quicker response time here with it getting to the appropriate pH. Now, when I do this type of experiment, I like to have some comparisons. So I'm going to actually just start with regular fresh water. And this is fresh water that I took from a fish tank. You can use any lake or stream water you might have access to. You can also just use tap water if you like as well. I recommend, though, trying to find a natural source of water, primarily because you get a lot of the more natural ions and other nutrients and stuff. So it's a little bit more of a realistic sample here. So I'm going to place my pH probe in my freshwater sample, and I'm making sure the glass bulb is submerged, but not touching the bottom of my beaker. And I'm gonna let it kind of sit in there and wait for it to reach a bit of an equilibrium state. And I can swirl this to kind of speed that up so you can see. Okay, and while that's going, I've already taken one Alka-Seltzer tablet and broken it up and put it into a 125 milliliter Erlenmeyer flask. And I'm going to take just 10 milliliters of distilled water and fill that up. So I am ready to go once my pH kind of reaches its equilibrium, which it's getting close. Some more swirling to kind of make sure I get there. It's getting close. I know that pH is closer to 7 of the sample because I've measured it quite a bit before. All right, are there any questions so far as I'm waiting to get this pH to get to equilibrium here?
Hey, Colleen, they're wondering if you could show them a closer view of the setup.
Yeah, no problem. Let me grab the camera here. Don't mind my arm. Okay, so I've got a 250 milliliter beaker that has 100 milliliters of my freshwater sample in it. I've got a Erlenmeyer 125 milliliter Erlenmeyer flask with one Alka-Seltzer tablet and 10 milliliters of distilled water. And then the way I'm going to transfer the CO2 is I have a stopper that has a lure lock on it. And this is actually, if you have Vernier gas pressure sensors, this is actually part of the accessory kit in those gas pressure sensors. And this is just the 2-hole stopper with a stopcock on one, so we make sure CO2 does not get released from there. And then the tubing on the other, that this is what I'm going to put into the sample like this to be able to actually bubble in our carbon dioxide. All right. And I can stop sharing here for a second, so it makes the picture a little bit bigger here so people can see a little better. So our freshwater sample, our Erlenmeyer flask with one Alka-Seltzer tablet, and then 10 milliliters of distilled water right here. And I think I'm pretty good on my pH there. Okay, so I'm going to share my screen again. Okay, now this next process is going to be pretty quick. What I'm going to do is I'm going to go ahead and start my data collection. And then as soon as my data collection goes for a couple seconds, I'm going to pour the water into my Alka-Seltzer tablets, stopper it, and put the tube into my water sample here so we can get the CO2 bubbling in it. So I'm going to start data collection. It's going to go for a couple seconds. I'm going to add water, this, and then add my bubbling here. And I'm just going to hold it. So, and then tend to swirl this a little bit to make sure that reaction is taking place and happening. And it took a couple seconds, but you can see immediately, oh, I've got my pH is now starting to drop. And I keep swirling it just to make sure the reaction keeps going. You want to make sure that stopper stays on pretty tight. And you can see the CO2 bubbling through. I'm just going to let that sit and keep bubbling slowly as the time keeps going. So like I said, it's a pretty quick reaction. So once you add the water, you want to act very quickly with that. And still going down a little bit. Looks like we're leveling out now. Okay. Now I'm just going to let it go for a few more seconds here before I just stop data collection. So I'm going to let go for a total of 120 seconds. And stop. All right. So what I can do is I can click on my zoom to all data and I can zoom on my data here. And in this lab activity, I'm gonna have my data table show up here. What I asked the students to do is actually determine what the maximum pH was and what the minimum pH was and determine the difference. Because what we're going to do is we're going to compare how much the pH drops in fresh water versus ocean water. And to help make sure I have my right data sets, I'm going to go ahead and rename my data set here so I make sure I know which is which. So I'm going to click on my data set options. And I'm going to go ahead and rename my data set. And this is going to be renamed freshwater. That way I know that is going to be my freshwater sample that I want to analyze. Okay, first run of data so far. Any questions? All right, so I'm going to go ahead now and get set up for the second run of data. So I'm going to remove my pH probe from my freshwater. I'm going to go ahead and rinse it off. Distilled water here. Light towel dry. And now I'm going to use my ocean water. So my ocean water, we are lucky enough that we actually have a saltwater fish tank here in the office. So I'm going to just take 100 mils of water from the saltwater fish tank. Put that again in so the glass bulb isn't touching the bottom. And kind of give it a swirl. Get up the appropriate pH. Okay, and as that's settling in, what I'm going to do is I'm actually grabbing a fresh 125 milliliter Erlenmeyer flask. And the reason I'm grabbing a fresh one is because the Alka-Seltzer reacts so easily, quickly with the water, is I don't want to have to clean and then really dry out that other Erlenmeyer flask. So I'm having a second one that's already dry and clean just to help kind of cut down on the time that this reaction is, or the time for this experiment to happen. So I have another Alka-Seltzer tablet, and what I made sure to do is I kind of crush them up here. You can pulverize them more if you want. It just means the reaction will go faster. I mostly crush them up because the size of an Alka-Seltzer tablet is bigger than the opening on the Erlenmeyer flask. So it's going to be hard to shove one in there all the way. All right. I'm going to do my 10 milliliters of water again. All right. A pretty good pH going here of my ocean water. It's kind of on point for what it should be. And again, I'm going to go ahead and start my data collection. Again, I'm going to let it go for a couple seconds. Then once I have it going for a couple seconds, I'm going to go ahead and add my water, stopper it, and put my tube in my ocean water and let it go. As it comes out. Here we go. All right. I'm gonna let this keep going. That's it. As it keeps reacting and going, you can see I'm getting the same kind of reaction happening here where my pH is dropping. So I'm going to go ahead while this is happening. I'm going to go ahead and put up my fresh water as well. We can see how the pHs are going to be very different from their starting pHs because freshwater pH does tend to be quite a bit lower than saltwater pH, just because of the different ions and everything present in this water samples. And I'm going to let that go the full same kind of time span here. Probably going to let it go a little bit longer this time because it's still decreasing a little bit more. All right. Okay, and then I'm going to rename this data set ocean water. Again, making sure I'm able to kind of differentiate between my two data samples that I've been collecting here. So now that I have my two pieces of data, what I can do now is actually share this data with you so that you can do some of your own analyses on it. So I am going to share my presentation again. And so to do this, what you need to do is activate Graphical Analysis Pro. So on your device that you have graphical analysis on, there should be a notification at the bottom of it saying that Pro is not activated. And then you want to enter this code, this code right here into that activation. And it's, Angie, if you could copy and paste that into the chat, that would be great. It is case sensitive. So I recommend if you're doing this on a computer that you just copy and paste it. And then once you have activated that, okay, I'm going to go back to sharing my other graphical analysis here. So what I'm going to do is I'm going to turn on data sharing. So I'm going to click on my data sharing icon and I am going to start a data sharing session. And so the code for this session is going to be DWR7IA. So you are going to have two codes, one to activate Graphical Analysis Pro, And that code is going to be good for you for 2 weeks. And then this other code, which is going to be what we actually are going to share the data over the internet with. And this code is only good for the time period that I am actually sharing your data. So you want to write this down because I'm going to show you where you're going to enter it. So under new experiment, I'm going to save this. Here, you're going to want to go to data sharing and enter the code DWR7IA into the online data sharing source ID. And then you're going to click connect. And once you've entered that code, you should have the data that I now shared with you. And I can actually see how many people are connected right now. And I can see only one person currently is connected, but that shows that one person is seeing my data. And so this is kind of cool because now I'm able to share this. If I was doing this as I'm doing right now as a demonstration, I can share the data with everybody to be able to do their own analyses and analyze it. Turn off my little warning there. Okay, we got 2 people connected, right? So one other aspect of this experiment that is not written in the procedure, but is one that I like to throw in there, is because a lot of times you have students who wonder, well, what do you mean excess CO2? Because there's always CO2 in the atmosphere. So if we were just to put atmospheric CO2 in here, it would totally be fine, or it would change still, right? So I like to do another run of this experiment where instead of actually adding excess carbon dioxide, I'm just going to use a normal aquarium aerating stone and basically just bubble atmospheric air into the sample. And you'll see here what's going to happen with that once I do that. So I'm going to go ahead and change out my water sample. So I'm still going to do ocean water. I'm just going to get a new sample. And this one, I don't have to worry as much about making sure I have a dry beaker because I'm not using the Alka-Seltzer tablets anymore. And this is I think a really helpful step to help students understand the difference between regular atmospheric levels of CO2 and then when we're talking about excess levels of CO2. So I'm going to go ahead and put let my pH get back up there. And I'm using just a cheap $5 aquarium bubbler that I got at Petco or PetSmart, someplace like that, that I use to do other experiments, mostly dissolved oxygen experiments. And so I'm going to get my pH back up to where it was. And what's cool is if you're connected, what you should see is the data coming across live as this is happening. So I'm going to put my aerating stone in. I'm going to hold this. I'm going to go ahead and do what I did before. I'm going to start my data collection. Let it go for a couple seconds. And then I'm going to plug in my aerator. And so the key here is thinking about this experiment that we're not adding excess CO2. All we're doing is putting in atmospheric air into here. So it's the normal level of CO2 we currently have. And yes, my pH is rising a little bit, but that's because I'm on limited time with y'all today. And I did this before my pH fully reached its equilibrium state. And if I pull up my other two graphs here, you can see right away there is a dramatic difference taking place of when I add excess CO2 versus when it's just atmospheric levels. And that's a key talking point when you're talking about climate change, to make sure you're expressing things in excess levels of carbon dioxide, basically levels that are not normal. And by showing, this is what atmospheric air does, but if we keep adding CO2, we're going above that normal level, and we're going to see excess CO2 having a dramatic effect on our ocean water samples. Go just full 2 minutes. All right. And then I can unplug it. And so we can see, looking at these, that just adding atmospheric air in there, those CO2 levels did not change the pH. The pH stayed stable during that time. Versus the times we added excess CO2, we're having more carbonic acid forming, and that's going to cause that pH to drop, which is going to make it more difficult for organisms to be able to make their calcium carbonate structures in order to survive and be viable, have viable offspring. Now, a couple of cool things here. So what I can do is I can actually do view statistics on this data, and this is going to actually give me my statistical information for each run of data. I can also do a graph legend, so I can actually see which is which on here based on the color. And the key things that we pay attention to in this activity, again, like I said earlier, are looking at the total change of pH that happens here. So we get our minimum and our maximum, but we also get our delta Y, so we can get our complete and total change that happens. What you should notice, for those of you who have tapped in and are getting my data with me, this information should not have transferred over, because when the data is sharing, it's only the data that's being shared out. It's not the actual analysis. So it requires the students to do their own analysis on the data themselves. So I can look at my difference here of, ocean water versus fresh water, and see I had a larger change in my ocean water pH and then in my freshwater pH. And so that's a great talking point with students of, okay, so this acidification process is going to affect freshwater as well as ocean water, but we're seeing it more of a dramatic effect on the ocean water. And you can go into some of the questions of why that might be placed, and why is this a bigger effect on ocean systems than freshwater systems? Or is it an effect on freshwater systems and it just hasn't been researched enough? So it goes off on a lot of great talking points that you can do about climate change, and specifically the effects that excess CO2 in the atmosphere is having on not just the temperature of things, but also on the actual chemistry, especially ocean chemistry, and talking about that process. So, are there any other questions?
There was one question, Colleen. Could they use instant ocean mix?
Yes. Yeah, so you can use instant ocean mix. There also are recipes out there that you can look up that are the different salts, because it's more than just sodium chloride. There's magnesium chloride, and I forgot the other salt that goes in there as well. In a pinch, you can just use a normal saltwater solution. You're probably not going to see as dramatic or as big of an effect if you compare it to fresh water. And same thing with fresh water, you can just use tap water, but again, it's not going to be as robust of an experiment in that case. But there are other recipes out there to do fresh water and salt water that are available. I believe in some of our books, we also have recipes for making your own for those cases. But instant ocean is what we use to actually make our salt water here. So it would be that I know will work great because that's just what I used. And oh, I see in chat, somebody asked if you could see who's actually connected to your data sharing. No, you can't. It only shows you here. I can share that here. It only shows me how many people are connected to it. So here it says 3 clients are connected. So this is a great option too, especially if you have students who might be out because they're sick and you might be doing a lab activity that day. If they're up to it, they can actually join in and have the data shared to them by their lab mates and be able to actually have the data to do the lab reports with that way. And I can just stop data sharing by clicking stop sharing. And that should turn off the data. But what's great is the data that's already been shared out is already on those devices. So you just can save the data onto your device and it's there for you to keep.
There's another question, Colleen. Can you export the graphs to Sheets or Excel?
So excellent question. So let me back on here. So over here under File menu, you can go to an export option. And from this export option is where I can actually export my graph. It will also export all the things on that graph as well. So you can see I would want to turn off my statistics and move my legend a bit out of the way. But from here, you can actually save it as a PNG file, which you can then just drop into a Google Doc or a Word document to be able to put it into a lab report. You can also export the data as a CSV file. So that would just be the data itself, not the graph. And then that CSV file can then be imported into other Sheets programs such as Google Sheets or Excel. Any other questions?
I have another question. Hold on here. Please address the possible disconnect that some data collectors may have when they reason out that it is the present atmospheric CO2 that is causing the acidification of the current ocean water chemistry. If the pump continued to run, would the solution in the beaker drop over time? I think what that means is, I think the question is, right, as the atmospheric CO2 is changing, it will change over time. At the ocean pH will change over time as the atmospheric CO2 changes. You're demonstrating that right now, this is what the atmospheric CO2 is.
Correct. Yeah, I'm kind of doing because even with us adding the excess CO2, the excess CO2 here is basically a sped up version of what's happening. It's more of a model than an actually like this is happening today where we're seeing a pH drop of 1.9 in our ocean water, because remember the pH logarithmic scale. And if you were to talk about the key here is that excess CO2. So the fact that we're at, I believe it's now 420 ppm And so as we keep increasing in our carbon dioxide concentration, more and more CO2 is going to be added to the ocean, and we are going to see that drop. But in this instantaneous point of time, we're just looking at the concentration of CO2 currently and looking at what happens when we add extra by doing the chemical reaction with the Alka-Seltzer tablets. And so in theory, if we were to keep that bubbling going for years, we would definitely see a decrease in pH because of the fact that more and more CO2 is constantly being added to the atmosphere. And so as CO2 concentration in the atmosphere increases, you're gonna see the drop in pH of ocean water. Great. Well, as you're kind of digesting all of this, if you have any questions that might come up later, don't hesitate to reach out and ask. You can reach me at biology@vernier.com. Angie shared a folder in the beginning that's going to have a PDF of this presentation along with the a PDF of the lab write-up. It's a bit of a rough lab write-up. I know some images would be nice to be added in there to help make things clearer. But if you have any suggestions or questions about it, please don't hesitate to reach out. This is just something I wrote up during pandemic times to have kind of a more interesting way to talk about ocean acidification. Thank you for joining us today, and I hope you have a wonderful Thanksgiving, and everyone have a great day. Thank you.